Hello everyone. Today we are starting with the program that is for Bachelor of Science second year Botany. Semester 4, paper code is BOC 104, paper title is Plant Physiology. Title of the unit is from the Unit 1 Plant Water Relations. Module name is Transpiration Part 1. Module number 5. I am Dr. Nisha Kewat from DPM Sri Malika Arjun and Sri Chetan Manju Desai College, Kankona, Goa. In this presentation, we will be covering about the transpiration, the various aspects such as introduction, kinds of transpiration, and the measurement methods of the transpiration. Learning outcomes. At the end of the session, a student will be able to know about the water movement, define the transpiration. They will also know the three kinds of the transpiration, and lastly, they will know the various methods that are available to measure transpiration. Coming to the introduction to transpiration, what exactly is a transpiration? If you see this plant, it's a schematic diagrams, the water is being absorbed through the root hairs, then water will be conducted through the conducting system, that is a xylem, and it will be circulated to the different parts. Then later on it will be utilized in the various metabolic processes such as photosynthesis, and the excess amount of water that remains will be released out through the stomata. Coming to the definition, there are various definitions that are available. Among that, these three definitions given by the three different authors are kind of an overlapping and they are similar to each other. For example, Miller 1930, according to him, the transpiration is a loss of a water in the form of a water vapor from stem and leaves. According to the Mayer 1953, it is a loss of water vapor from aerial parts of the living plants. Whereas according to the Thomas 1956, it is giving off of a water vapor from the surface of the plant. Coming to the demonstration of transpiration, if one would like to know how exactly or whether the transpiration, whether they exist or not, so this is a very simple experimental design that we can understand about the transpiration. You take a potted plant, cover the pot completely and the soil with a polythene bag. After some time, you seal the glass jar with the Vaseline so that it is an airtight situation which is created. After some time, we see that there is a water molecule that are accumulated around the plant area, which suggests that water has been transpired or water has been released out through the aerial parts of the plant. In this case, it is the leaves and a small green tissues. Coming to the kinds of the transpiration, in general, there are majorly three kinds of transpiration. The first one is cuticular transpiration, then lenticular transpiration, and a stomatal transpiration. Cuticular transpiration, it is the transpiration which is observed on the surface of a stem and a leaves. If you see this plant, I have taken an example of a xeropoietic plant, that is Nerium. If we take a oblique section, we can see the cuticle here and there is a loss of water from this tissue. But cuticular transpiration contributes to about only 10% of the transpirational loss. Coming to the lenticular transpiration, it takes place through the lenticell. This is a microscopic image. It is taken place through the lenticell, which is usually found on the secondary growth or on a woody stem, and it contributes very negligible amount of transpiration. Coming to the third type of a transpiration, that is a stomatal transpiration. Stomatal transpiration basically occurs through the stomata. If you see in this figure, this is the guard cells, this is a stomatal opening. From this stomatal opening, there is a gaseous exchange as well as the water movement is towards the outer side. So the water vapor is released through this stomatal pore. And the stomatal transpiration basically contribute around 90 to 97% of the transpiration. Coming to the methods of transpiration, there are overall four methods by which we can measure the transpiration. One is a weighing method, second one is a cobalt chloride method, third one is a collecting and weighing transpired water, and the lastly potometer. Coming to the weighing method, in this is a very simple experimental diagram wherein you can measure the transpiration. We take a healthy plant, potted plant, we weigh them, then we cover the pot completely with a polythene bag. Later on, after some interval, we weigh it again in order to analyze if there is any weight difference. We note down the weight of the plant Again, after some particular time interval, again we weigh the potted plant and we see the weight difference. So from the 0 day till the last day, whatever the differences that we get into the weight of the potted plant, we can indirectly relate it to the amount of water loss through the plant. Second one is a cobalt chloride method. In this case, we take a healthy leaf, then we cover it with the filter paper which are soaked in a cobalt chloride solutions which are blue in color. In this case, we compare the amount of transpiration which is taking place on the above surface of the leaf and the lower surface of the leaf. We see that after some time, above surface, the filter paper which is placed above surface, it has turned pink, but it is in a lighter shade as compared to the filter paper which is placed on the lower surface of the leaf. This observation suggests that the transpiration has taken place to a greater extent onto the lower surface as compared to the above surface. Third method, collecting and weighing the transpired water. In this case, we take a potted plant, we cover it with a bell jar, and then we pass a gas with a known moisture content. After that, we connect the bell jar with an anhydrous calcium chloride and we weigh it and we connect it. So after some time, we see that we take the weight of this anhydrous calcium chloride, which has become hydrated now because there has been a transpiration that has taken place by this potted plant. So the weight difference between the anhydrous calcium chloride and between the after weighing the calcium chloride which is hydrated, we can actually relate it to the amount of a water loss. Coming to the potometer method, there are 5 types of the potometer method by which the transpiration rate can be measured. The first one is the Darwin's potometer. It is the most simplest type of a potometer. Coming to the farmer's potometer, in this potometer method, we have a glass bell jar which is covered with the cork having 3 holes. In one of the hole, there is a bent glass rod which is inserted in this glass jar, and then the same is connected it is placed in another beaker which is containing water. In the middle, there is a fresh twig which is cut only in the water. Then in the third hole, we have a water reservoir which is adapted with a cork, like a stop cork. So we see that since it is an airtight container entirely, we see that whenever there is a water movement, whenever there is a water loss through the transpiration, there is a vacuum that is being generated in this jar. So in order to fill up this vacuum, the water will be taken up from this beaker. So we see here the air bubble which is placed over here, it moves towards this direction. So when it moves towards this direction, we can measure the ruler. With the help of a ruler, we can measure the distance that is traveled by the air bubble, and indirectly we can relate to the water loss. Gannon's potometer is similar to the previous one, that is farmer's potometer, except that instead of a transparent water solution, here it is a colored solution. Whenever there will be a vacuum generated, the colored solution, the colored water will be taken up and the air bubble will move this side. Coming to the Garros potometer, in this case, the modification from the previous two is that in this case, the above surface and the lower surface of the leaf, the transpiration rate is measured separately. There is a glass jar which is having a tube containing anhydrous calcium chloride, as well as on the lower surface there is a tube containing anhydrous calcium chloride. After some time, we take the weight of this calcium and anhydrous calcium chloride, and we see the difference in the weight. If transpiration has taken place, then definitely the weight of the calcium chloride will increase, and likewise on the lower surface. Coming to the Bose's potometer, here this is a glass jar. Again, it is airtight. We have a twig, we have a glass tube which is having a 2 bulb, that is the inner bulb and outer bulb. In an outer bulb, there is an oil drop which is being placed. So whenever there is a transpiration taking place, there is a water which is being taken up and it is released out. So again there is a vacuum being created, and in order to fill up that vacuum, this oil drop will move towards the through this horizontal tube, and it will go on top and it will burst. Likewise, when it bursts, then it will move down backward in the horizontal tube. So this process will continue, and the time which is taken between the bursting of the two oil drop can be calculated and related to the transpirational loss. In this case study, what we understand that whenever the potted plant is placed in the sunlight, we see that the movement of the air bubble, which is through the capillary action, it moves to this much distance, whereas in a shade it is 2.5, whereas in a darkness we do not see any change in the movement of the air bubble. Whereas when the plant is placed in front of the fan, there is too much of water movement. So in general it is says that transpiration is affected by the various external factors. So there are few misconceptions which are related to transpiration. That first of all, water absorption is a compensatory mechanism for transpiration. Second thing is that it is the only way that water is used by the plant for transpiration. And thirdly, transpiration is a loss of a water vapor from different parts of the plant. These are my references. Thank you.